Hello? Oh, she turned off. There. Is that on? There we go. Okay. So this is a very cool program. I really recommend it highly. I think it's an opportunity for everybody to not only get exercise, but also to see. It gives you a challenge to see how much you're exercising and improve on that. So that's a very good thing. Okay. Well, we turn our attention now to biochemistry. And we've got some distance to cover here. We got into membranes last time, and we're going to get through that today and talk about membrane transport, which is something we're going to focus on for most of this week. Membranes, as I noted last time, are the cell's barrier between them and the rest of the universe. And that barrier is a fairly impenetrable one for most molecules. As I noted, there were 4 small molecules that move across the membrane fairly readily, but most molecules do not move across the membrane very readily. I talked about proteins that were either embedded in or associated with or anchored or some means of association with the membrane. And one of those that I talked about was prostaglandin synthase relative to the prostaglandins that I was talking about. We'll talk a little bit more about prostaglandins later when we talk about their metabolism, and so you can learn something about some of their physiological effects. One of the things I noted was that there are molecules that are known as non-steroidal anti-inflammatory drugs, or what I call NSAIDs, N-S-A-I-D-S. And what these drugs do is they inhibit the action of prostaglandin synthase. So they inhibit the production of prostaglandins. The prime example of these type of drugs is aspirin, also known as acetylsalicylic acid, which you don't need to know that name. But aspirin you do need to know, because what aspirin does is it completely inactivates the enzyme by binding covalently to a serine within the enzyme. And that serine is involved in the enzyme's catalysis. It stops the enzyme from functioning and therefore stops the production of prostaglandins. So we'll talk more about those later in the term, as I noted. Okay. Now, one of the things that we see if we think about the structure of a membrane, we realize, oh, actually, there's something I should show you before then. So there is, when we look at protein structure, we discover something interesting. So one of the things about protein structure that I mentioned earlier last term was the fact that proteins are arranged so that the nonpolar parts of proteins are generally located on the inner part of the protein, and the polar portions that interact with water are on the outside. And that's true for proteins that are found dissolved in the cytoplasm. It's not commonly found for proteins that are found, for example, embedded in the cell membrane. And that sort of makes sense because if you think about it, looking at this protein that is known as porin, this guy is embedded in the cell membrane, and when we look at the portions that are embedded, they're interacting with the nonpolar tails that are there. So it wouldn't be too surprising to discover that for these embedded proteins that we might find some hydrophobic amino acids on the outside of these proteins. And in fact, that's exactly what we find. Okay? So proteins that are embedded in membranes have some specific structures that are kind of interesting. Okay? So one of the things that we see is that in going, this actually, I thought it was a figure, but it's not the figure. In going from the outside, across the membrane, inside, back across the membrane, you can see they're sort of going up, down, cross, and so forth across this membrane. We might expect that those portions that are associated with these green parts are nonpolar side chains, and these portions that are associated up here with the pink or the cytoplasmic parts would be more hydrophilic. And in fact, that's exactly what we see. So one of the things that we can do is we can predict for a protein its likelihood of being found in a membrane. Oftentimes we discover a protein and we don't know where it's found. Okay? Where is it? So we can analyze the sequence of that and say, okay, look, here's a stretch of 7 or 8 amino acids that's fairly nonpolar, and then it's got 3 or 4 that are polar, and then 7 or 8 nonpolar again, and we see this sort of repeating pattern. And those patterns are reflecting what you're seeing here. A portion that's crossing the membrane being nonpolar, and once it gets across, polar, and then coming back, nonpolar, polar, and then back nonpolar, and then polar, etc. So these transmembrane helices that we're seeing in proteins will give us a clue. If we see this pattern of nonpolar, polar, and so forth, it gives us a clue that this protein might well be found within a membrane. So that's very useful for us. And here's a good example here. Okay, so here's a protein that's found in a membrane. We see this transmembrane helix, and they're shown in yellow. And these yellow amino acids that you can see here are all very hydrophobic, meaning they don't like to associate with water. And again, they're not going to find water here in the lipid bilayer, but the portions that are on the outside or the inside or the outside of that membrane are fairly polar. And that's because these parts are interacting with water. So this really, again, the sequence of a protein can tell us something about the location we find it within a cell. Okay? And this is simply a plot of the hydrophobicity, that is the tendency to be hydrophilic versus hydrophobic. And as you can imagine, this would change every time it crosses a membrane. Okay, so sequence of a protein is very useful for us to help us understand, in fact, what a protein is, where a protein is actually located. Well, thinking more about membranes, there are some other things, some other considerations for membranes. Membranes are one of the guiding principles of membranes I want you to sort of associate with is that membranes are what we call fluid, meaning that they're not a fixed structure. Okay? Like a fluid, they move, they flow. Okay? They're not solid structures unless we get them too cold, and I'll talk about that in a little bit. Okay? They're not very cold. Okay? This figure illustrates one of the ways in which people study membranes. All right? So they can, for example, take a cell, and that cell has a nucleus, it's got various things, but using a laser, they can bleach. They can actually use a laser to bleach if they've colored the cell, let's say, with a dye or something. They can bleach that cell in a very specific place, like you see here in B. So this white circle is a place where a laser has changed the color of that cell. If we let that laser, take the laser away, and we see that color, and we watch how long it takes for that color to disappear, we get an idea of the fluidity, the rate with which things are moving in that membrane. And it turns out they're moving pretty fast. They're moving pretty darn fast. So that, again, is consistent with what people call the fluid mosaic model of a membrane. So a fluid mosaic model of a membrane simply says that a membrane is not a solid structure and that it, in fact, is very fluid. Okay. Now, when we see molecules like the glycerophospholipids or the sphingolipids within a lipid bilayer, we might ask the question, is there a bias, first of all, that we tend to find, let's say, with respect to a cell membrane? Is there a bias where we expect to see maybe more of one type of glycerophospholipid or sphingolipid on the outside compared to the inside? And it turns out that for many membranes, we actually do see a bias. There is a bias. And it's not completely clear in every case why there is, in fact, in many cases, it's not clear at all why there's a bias, but that bias appears to be important for maintaining stability inside of a cell. Okay? How does that bias get made? Well, it gets made in two ways. One is if the membrane is, as it's being made so that only certain molecules are put on the outside versus the inside, then that would obviously determine a bias. And the other is that once a membrane has been assembled, it's possible to flip the components of a membrane. Okay? So flipping a component of a membrane is known as transverse diffusion. So I'm going to show you, it's the bottom one here. I'll talk about it first. Transverse diffusion means that this guy here, who is in sort of a darkish pink, is moving from the top down to the bottom. That type of movement happens extraordinarily slowly by itself. It doesn't happen very rapidly. Okay? It takes a long, long time for that to happen just randomly. Cells have enzymes called flipases, F-L-I-P-A-S-E-S. And flipases will actually catalyze what you're seeing there. Well, the fact that cells have enzymes that catalyze this transverse diffusion tells us that the arrangement of these glycerophospholipids and sphingolipids as to whether on the inside or the outside is important, even if we don't really understand why that's important. Okay? The other type of movement that we see in membranes is much more common, and in fact, it's actually what we see with respect to the fluid mosaic model, and that's called lateral diffusion. Notice that in lateral diffusion, this guy, the dark one, is moving simply from left to right. It's staying on the same side of the membrane, but it's changed its position with respect to the other molecules within that membrane. Lateral diffusion happens very, very rapidly, and that's in fact why we see the fluid mosaic model actually makes a lot of sense. Okay. And let's see. That's just simply showing you graphically what the experiment was doing there. All right. Well, another thing we can think about with respect to membranes has to do, again, with their fluidity, but now we're examining their fluidity as a function of the temperature in which they're located. Okay? So we can think of a membrane as being very fluid, meaning a lot of movement, a lot of back and forth, a lot of lateral diffusion, very rapid lateral diffusion. And we can imagine that if we took that membrane, or took that cell that has that membrane, and we started to cool it down, like anything that you cool down, you would expect that that movement would start to slow down, and you might have some different characteristics of that membrane. Well, in fact, what happens is the membrane does physically change its nature of interaction. It really does physically change. We can think of that as being more like it's frozen as it's getting colder and colder. Freezing is not a good example because freezing we associate with crystallization, like with water and so forth, but freezing making it more solid-like and less fluid-like. Okay? So this graph shows a membrane that if we start at a very low temperature and we start increasing the temperature, we see it goes through a transition from being more solid-like to being more fluid-like. Okay? The midpoint of that transition is known as the TM. Some people call it the melting temperature. Okay? But it's really a transition from one state to the other state. Well, it turns out that fluid membranes are really important for cells. Okay? You want your cell membranes to be fluid. As they start to solidify, cells can't interact properly, and there's all kinds of problems associated with that. And it turns out that there are some factors that influence the fluidity of a membrane. Okay? Some factors that influence the fluidity of a membrane. Okay? Here are some phosphatidylcholine containing different pairs of identical fatty acid chains. And we look at their TMs. What do we see? Well, we see, first of all, that the shorter the chains are, the more the melting temperature goes down. Okay? So shorter chains are consistent with fluidity at a lower temperature. Longer chains tend to cause the melting temperature to increase. But more importantly than the length of the chains of the fatty acids within this phosphatidylcholine is the unsaturation. Look what happens when we put just oleic acid in there. Oleic acid has one double bond, and oleic acid drops the temperature of that is the TM, the melting temperature, down to minus 22. Okay? It tells us that unsaturation has a very dramatic impact on the fluidity of membranes. The more unsaturated the fatty acids are that are found in a membrane, the lower the melting temperature will be. Now, this makes a lot of sense, and it makes a lot of sense from a nutritional point of view. How does it make sense from a nutritional point of view? Well, think about, for example, a fish swimming out in the ocean. Or think about a plant that's out in a very cold environment. Both of those are in circumstances where they would be, if all they had were saturated fatty acids in them, their membranes would cease to be fluid in the environment in which they were located very quickly. Okay? These systems have evolved to use fatty acids that are more unsaturated in making up their membranes, so their membranes can stay fluid in the environment in which these organisms are found. It's not surprising that plant oils are rich in unsaturated fatty acids. It's not surprising that fish oil is rich in unsaturated fatty acid because they're coming from organisms that have these properties in these environments in which they need to exist. Okay? So this is why fish oil is unsaturated. It's why plant oil is unsaturated and so forth. Yes? How do double bonds contribute to the fluidity of the membranes? Thank you for asking that question. And the answer is actually shown here. Here is what, this is actually showing you for a fat, but the principle is the same for a glycerophospholipid. Here is the stacking that we see for a saturated fat. Okay? Stacking, the more things get stacked, the more solid-like they are. All right? It's harder to keep this fluid. We actually have to heat it up to keep the things moving around and so that it remains fluid. If we put a double bond in there that has a bend in it, look what happens. It's much harder to get this guy to stack. And in fact, it takes a much lower temperature before these interactions that are here that are giving fluidity will start to dissipate. Okay? So it's this bend that's in the unsaturated fatty acids that disrupts the organization and consequently disrupts the fluidity, increases the fluidity of the organisms that contain this in their membranes. Okay. Cholesterol, I said earlier, was very important for membranes, and I didn't tell you why. Okay? I said that cholesterol was a very prominent component of the membranes of our brain, for example, 14% of the dry weight. And cholesterol is made in the body so it can be put into membranes. It's also made so that we can make steroid hormones. Okay? And bile acids, that's another thing that we make from it. But from a membrane perspective, what cholesterol does is it fits in membranes kind of like what you see here. And the organization of cholesterol in the membrane causes the property of the membrane to change yet further. Cholesterol does not, underline, does not lower or raise the melting temperature. It doesn't change the melting temperature. Instead, what it does, if we go back to the, if we go back to the graph that I had here, what it does is it widens the transition range. Okay? So instead of this being a fairly steep rise right here, what cholesterol does is it causes it to start rising over here and making this much more of a gradual incline. Again, not completely clear why that's important, but that's what cholesterol does to membranes. It widens that transition range of membranes. And it is important because cells make a lot of cholesterol to put into their membranes. Okay. Now, I want to introduce this. We're going to talk about this later today, actually, as well. But this is a something that the first example that we see of movement across a membrane using a protein. We've talked about how water can move on its own. Carbon dioxide, oxygen, carbon monoxide can move on their own across the membrane. But cells have many things that they want to move across membranes. One of the things they want to move across membranes are sodium and potassium. It turns out these two are really critical. Really critical. Okay? I'll tell you why. All right? So there's a protein called the sodium-potassium ATPase. That's what you see on the screen. The sodium-potassium ATPase is a protein that's found. It's an integral membrane protein. You can see it here. And this integral membrane protein does the following thing. It performs something we call active transport. And active transport means that something is being moved from an area of lower concentration into an area of higher concentration. We're moving against the concentration gradient. So this protein is performing active transport. The analogy I like to use is thinking about, well, let's imagine that I have a whole bunch of water sitting down here, and I want to get that water up to where these guys are up in the balcony. All right? Water isn't going to go there all by itself. I'm going to have to put some energy in to make that happen. And similarly, I must use energy to move against a concentration gradient. Okay? So active transport processes take and require some type of energy. In this case, you can see the energy is ATP. ATP is not an absolute requirement. Some protein systems do it without ATP, but in this case, ATP is used. What this protein does is it goes through a cycle. We'll see the cycle later. But the cycle, during the cycle, there are 3 ions of sodium that get pumped outwards, and then 2 ions of potassium get moved inwards. Okay? Why do we care about that? Well, let's think about it. Sodium and potassium are ions. If I put 3 out and I bring 2 in, am I changing the charge? You betcha. This pump is what we call electrogenic, meaning it creates a charge difference as a result of its action. We also call this protein an antiport, meaning that I have molecules moving in opposite directions from each other. 1 out, or 3 out, 2 in. So out versus in, right? Why do cells do this? Well, cells do this because they have to. They do it because they have to. Why do they have to? Let's imagine a cell sitting here in my skin. This cell is full of proteins. It's full of carbohydrates. It's full of, hopefully not too much fat, but it's full of all kinds of things that are not on the outside of that cell. How many of you did the experiment in basic biology where you took some dialysis tubing, you put some protein in there, and then you tightened it up and you watched what happened? How many did that? What happened? Osmotic pressure. Water flowed in. And why did water flow in? It's trying to equalize the pressure, but it can't equalize the pressure because the protein can't get out. So it keeps trying to dilute it and trying to dilute it, and if you let it go long enough, the bag will actually burst. Okay? Cells have the same problem. Cells have the same problem. They're a bag of things that can't get out, and you've got all this water around them. If you don't do something to adjust the osmotic pressure, cells will burst. They will burst. This is an attempt to balance the osmotic pressure. Now, it's way more complicated than we're going to go in here to do it, but you can imagine as we're changing ionic concentrations, we are changing the ionic environment that's around this cell. And if we do it intelligently and we do it properly, we might stop that movement of water, because remember, water can come in all by itself just fine. And that's exactly what happens. Cells are doing this because if they stop doing this, they will burst. This is one, you hear about the cost of living? This is one of the costs of living. It's ATP. Okay? ATP used right here is one of the costs of living. You don't do this, you got serious problems. Now, we're going to see that cells, being the opportunists that they are, use this system to do things like send nerve signals. They use it to drive other pumps and so forth. So we're going to see some examples of both of those. But for our purposes right now, we're seeing 3 sodiums out, 2 potassiums in. Okay. And by the way, I sort of said it, but I'll make it clear. As you see here, there are more sodiums on the outside than there are on the inside. That's why it's taking more effort to continue to push more sodiums out. There are more potassiums on the inside than there are on the outside, and that's why it takes more effort to continue to push the sodiums, or pull the potassiums in, I'm sorry. Okay, so, let's see, bacterial membranes. Bacterial membranes are more complicated than our membranes are. They have layers to them. They have some covalent bonds in them. And bacterial membranes, you might remember from last term, bacterial membranes have unusual amino acids within them that are part of peptides, and those peptides sort of help to hold the membrane together and give some integrity to it. Those amino acids are in the D configuration. It's one of the few places we find D amino acids. Those D amino acids in those peptides can't be attacked by proteases. Bacteria are protected as a result of that. Okay. There's other membranes, beautiful things that are there. Okay. Eukaryotic cells are different from bacteria in that they have internal organelles, and each of those internal organelles have their own membrane. Each one has its own membrane. The nucleus has its own membrane. The mitochondrion has its own membrane. The endoplasmic reticulum has its own membrane. A lot of membranes in eukaryotic cells. Okay. And the last thing I want to say is actually right here. I'm going to talk to you about the ways in which we move things across membranes using protein pumps, like the sodium-potassium ATPase. But that's not the only way that we can move things across membranes. What you see on the screen is another way of moving things across membranes. It's called receptor-mediated endocytosis. I want you to write that down. Receptor-mediated endocytosis. This is the way that things like cholesterol make it into our cells. Cholesterol makes it into our cells by this mechanism. Okay? We'll talk about LDLs, low-density lipoprotein complexes, that carry lipids like cholesterol. And those LDLs come into the cell by binding to a specific receptor on the cell, as you can see on the left. Then the cell sort of pinches off and pulls that LDL in, and you can see it has now been internalized inside of the cell. This mechanism is yet another way of getting things into cells. Okay. Well, we'll talk about that mechanism later in the term, but for right now, what I want to go back and talk about is more about membrane transport itself. Okay? So let's review a little bit of that from what I've already said about membrane transport, and then we'll look at some specific examples. Okay? What does this graph tell us? It tells us that on the left side, we've got a change in free energy, and that is the y-axis, and on the x-axis, we have concentration ratio. It tells us basically something very simple. I already told you. And what I told you was that if we try to push something against a concentration gradient, it's going to take energy. Conversely, it tells us that things will tend to move from a high concentration to a low concentration if you leave them all on their own. You know that as entropy. Okay? You also know that as diffusion. If I take a glass of water and I take a drop of dye and I put it in the glass of water, I see it go in as a drop, but I come back 15 minutes later, and now it's completely mixed. It went from a place of high concentration at the point of the drop, and it diffused outwards and mixed completely upon, thanks to the process of diffusion. Okay? All right. I'll come back and talk about that. Okay. So, When we think about movement across membranes, there are, as I said, proteins involved in almost every case. Even in the receptor-mediated endocytosis, there are proteins that are grabbing onto those LDLs. For the things we're going to be talking about today and a little bit on Wednesday, the proteins are actually providing channels for the movement of materials through them. The proteins are providing channels for the movement of materials through them. What you see on the screen is a very simple kind of channel. Okay? The blue protein that you see is an integral protein. And the blue protein has a specific size and shape that will generally only allow very specific molecules to enter. This molecule might be, for example, glucose. Our blood cells have a protein like this that allows them to get glucose fairly readily. It will only take glucose. They're specific enough that this protein won't take, for example, galactose, which is very similar to glucose. So they're very specific at what they will accept. In the case of this protein, you can see that the binding of the glucose, the red molecule, in the receptor, in the transport protein, I shouldn't say receptor, but in the transport protein, causes the transport protein to grab a hold of it and change shape. And the grabbing a hold of it and changing shape causes the glucose to move from outside to inside. This process I've just shown to you doesn't require any energy. It will be driven totally by the process of diffusion. If the concentration of glucose is higher on the inside, it'll go backwards. It'll move upwards. If the concentration of glucose is higher on the outside, it'll move inwards. Well, for blood cells, this turns out to be really good, because in the fluid around the blood cells, there's generally more glucose than there is inside of the blood cells. So the cells don't have to waste any energy bringing glucose in. Okay? It allows the glucose to come in without energy. Not all things work that way. Okay? Not even all glucose transporters work that way. But this is one that doesn't require energy. And it works because the concentration of the glucose in the space between the blood cells is higher than it is inside of the blood cell itself. Okay? Well, here's a protein that also requires energy. Okay? Like the sodium-potassium ATPase, the calcium ATPase that you see here requires ATP energy. Okay? You remember last term, at the very end of the term, where we talked about the endoplasmic reticulum having all this calcium stored inside of it? This is one of the ways that all that calcium gets sequestered. Okay? One of the ways in which it can get sequestered. It's also a way in which cells can bring calcium into them, because calcium is obviously necessary for cells. Okay? This is an example of a protein that's in a class of proteins that uses an intermediate called phosphoaspartate. You see the aspartic acid, number 351, that's the position in the protein. During the movement of calcium into the cell, or into the organelle, that doesn't really matter, ATP, you see, binds, and during this movement process, a phosphate gets transferred to that aspartic acid. That causes two things to happen. One is ADP can leave, as you see here. It causes the protein to change shape because we've changed the charge of the protein. And that change in the shape of the protein causes the calcium ions to now be moved. Okay? Causes the calcium to be moved. All right? Calcium has now been moved across the membrane. All right? The rest of the process involves the removal of the phosphate from the aspartic acid. That simply is a hydrolysis, as you can see going on here. And when the phosphate gets removed, the protein goes back to its original shape, and now it's ready to go repeat the whole process again, binding calcium and going and doing the same thing. The process is driven by the hydrolysis of ATP. Okay? ATP makes this happen. Okay. Yes?
In that last step, it's step 4.
Yes? So for our purpose, we don't say outside or inside, but it's moved across the membrane. So that don't confuse yourself with that. Just it's moved across the membrane. Okay? All right. Here is another one. This one actually is not in your book, but I like this figure because it shows us how a sodium gradient can be useful. What is a sodium gradient? Well, whenever we talk about a gradient, we're talking about things that have a difference in concentration. So the sodium-potassium pump, when it works and works and works and works, it puts a lot of sodium out and it puts a lot of potassium in. And that gradient, that difference in concentration, means sodium really wants to come back in. 2 reasons. One, the sodium concentration is greater outside than inside. And second, the charge. Remember, we've been changing the charge of this guy. So there's more positive charge outside than inside. So these positive ions would like to come back in the cell. There's actually an electrical potential across the membrane. Okay? So cells have spent a lot of energy building this up. If they let a little bit of that sodium back in, they can do it without upsetting the osmotic balance, and they can use the energy of that sodium's movement to bring something with it. Okay? Well, the something that it's bringing with it is glucose. This is known as the sodium glucose pump. Here's a place that's using active transport. It's a system that's using active transport. It's not directly using ATP. It's using a gradient of sodium to bring in glucose. Remember that in active transport, there's always, underline always, always at least one molecule that's moving against a concentration gradient. In this case, the thing moving against the concentration gradient is glucose. Sodium is moving with the concentration gradient, meaning it's moving from high concentration to low. That gradient is made by the sodium-potassium ATPase. Okay. I'll stop there and take questions. Yes?
Is the osmotic pressure determined by what type of molecule is there, or just by what charge it has?
Yes. Her question is the osmotic pressure determined by the type of molecule that's there or the charge? So it's actually a combination of both things. Okay? So osmotic pressure is actually quite complicated when we think about all of the different things that are there inside the cell. But the type of molecule, the charge, both will affect the osmotic pressure. Okay. Everybody understand how the glucose is making it in? Yes?
Do we always see the sodium glucose pump coupled with the sodium potassium pump so they can create that?
So his question is, do we always see the sodium glucose pump coupled with the sodium potassium pump? This figure makes it look like they are coupled, but in fact, this is an opportunistic pump. However sodium got out there, it's going to drive the movement of glucose in. So it's not absolutely coupled, no. Okay? But in reality, the most likely way that it got out there was by action of the sodium-potassium pump. But remember, this is cell to cell. This sodium right here could have gotten there from a different cell, right? So it's not like it's one's feeding the other directly, right? Yes, back in the back. Okay. His question is providing that sodium gradient for this pump the only purpose of the sodium-potassium pump? No. The sodium-potassium pump has many purposes, the primary one being it's avoiding the cell bursting. It's maintaining the osmotic pressure. So that's why I say we don't want to do this too long, or we're going to have the same problem as before. So we want to make sure we do a little bit of this We use that to some extent, but not absolutely, or otherwise the cell is going to burst. Okay? Yes?
Would the sodium glucose importers have hydrochloric amino acids on the inside and hydrochloric on the outside?
Okay. So his question was, what would the organization of the amino acids in the sodium glucose one, or sodium? Okay. So, well, it's a little complicated, but on the outside they will certainly have nonpolar amino acids wherever there's a cross-membrane, a transmembrane domain. Yes. Okay? Yes?
So, for example, we know that ATP, those sodium potassium pumps, they work all the time, right?
Yes.
And given, for example, something happens and sodium cannot get back to the cells,
Yes.
lead channels, those channels, is it possible that the cell will pretty much just shrink?
Okay. So his question is actually another good question. So his question is, let's say sodium can't come back into the cell, and if this pump keeps working and working and working, is it going to cause the cell to shrink? No. In fact, what happens, what we see is as the concentration gradient gets high enough, just like as if I start putting a lot of water up here, after a point I can't push any more water up there, it's going to stop. And this is going to stop when the concentration gradient gets too high as well. It's a very important point. That gradient has a limit. It's not like it just keeps pushing it out blindly and nothing happens, but that concentration really has an effect. Just like the more water I put up there, it's harder for me to push more water to the top. Okay, very good. Yes, Kimberly. Oh, that's a very good question. Can we use these pumps to create ATP? We'll see that the vast majority of the ATP in our cells is made by a pump, not this one, but a pump that's kind of like this one. Okay? Yes, we absolutely do that. Yes, where you at? Oh, okay. If you're running an ATP deficit, will your cells pop? Okay. Well, obviously, I'm not running enough to pop too many of my cells. I like to run. But you have to keep in mind that cells have some pretty good resources for making ATP. We like to think of the fact that, well, I'm just so out of energy, I have no ATP. Your ATP concentration, when you're so out of energy, probably dropped 5%. Okay? So you still have a lot of ATP that's there. Thank goodness for that, right? Okay. Good questions. All right, let's think about some other things relative to human health. Digitoxygenin. Here's a really interesting molecule. It's a poison, and it's a poison that we use for heart patients. Not because we dislike heart patients, but because this poison in very tiny amounts actually turns out to be very useful. Why? Well, what it does is it knocks out the sodium-potassium pump. It knocks out the sodium-potassium pump. Okay? Let's see, where am I at? That's not what I want. Oh, blast it. Okay, well, I thought I had a figure, but I'll have to explain it to you. I tell you, maybe I'll save that for next time. It's going to take me a minute to explain. Let me give you a few more terms, and then I'll come back and talk about digitoxygenin next time. Okay? Symporters, antiporters. I talked about what an antiporter is. It moves molecules in opposite directions across a membrane. The sodium-potassium pump is an example of that. The sodium-glucose pump is an example of a symporter. Okay? Example of a symporter. A symporter moves two molecules in the same direction. I will not use the term uniporter because I think it confuses with an antiporter, so we won't talk about uniporters. Okay? Here is another pump called the lactose permease that uses protons to move things across a membrane gradient, across a membrane. So it's a proton gradient to move things across a membrane. We'll see on Wednesday how a proton gradient can actually get made. It gets made, no surprise, by pumping things one direction, in this case protons. And so if you pump more protons out, you've got more protons out that want to come in. And that concentration of protons, just like we saw for the sodium glucose, can be used to bring something in. In this case, bacteria use this to bring in lactose. How do they do it? Well, look at this. This is kind of a cool system. Here's the positively charged proton. Here's a carboxyl. It's bound to the carboxyl, and what has it done? It's changed the charge, and what does changing charge do? You know by now that it's changing the shape of the protein. Lactose is able to bind as a consequence. It binds, and the binding of lactose causes the protein itself to change again. Now the protein bends inward, and the lactose is in. Okay? The inward environment is more conducive to taking protons than the outward environment was because, remember, there's a gradient. The protons want to come in. The proton leaps off of the carboxyl, okay? And there we are. All right? Now, this is actually a mistake. Why this lactose is not supposed to be here. This lactose should be down here someplace, okay? And we're back to where we started. But the point is that, again, you don't see any ATP being used here. You're using simply a gradient to bring the thing in. Yes?
Well, so we have it's deprotonated, then it's protonated again, even though the proton is already inside.
It's getting protons from the outside.
Oh, it's another.
That's right. So we have a gradient. We have more protons outside than inside, right? Okay. I was going to sing, but I think, what do you say we call it a day? Yeah, let's call it a day. Okay.